My name is Karen Furie. I'm chair of the Brain Health Scientific Subcommittee of the Stroke Council. I'm here today with Dr. Kimball to discuss her study looking at cellular aging and the risk of stroke, dementia, and depression. Dr. Kimball, can you talk a little bit about the trial that you're presenting today?
In our study, we wanted to understand if leukocyte telomere length was associated with age-related brain diseases. And as you mentioned, we wanted to include stroke, dementia, and late life depression, as these three diseases are one of the most prevalent age-related brain diseases, and they also tend to co-occur, and they compound each other's risk, and they represent a very high burden for the patients and their caregivers.
What is a telomere?
So the telomeres are the protective caps of the DNA, which are in charge of protecting our cells from damage.
And you found that the length of the telomere was important. Can you explain a little bit more about that?
Yes, so as I mentioned, telomeres represent they are a marker of biological aging. So they represent the cellular aging that our cells go throughout the lifetime. And in our study, we identified that individuals that had shorter telomere lengths were associated to an increased risk of age-related brain diseases, which include stroke, dementia, and late life depression, individually and also as a composite outcome.
And the fact that the telomeres are short, is that a genetic situation?
That's a very good question. Telomere length is highly influenced by modifiable and non-modifiable risk factors. So yes, genetics plays an important role in telomere length. It is estimated that telomere lengths have an heritability of up to 80%, but also something important to take into account is that it's also highly influenced by lifestyle factors. So it also depends on lifestyle factors and also environmental exposures. And of course, different studies have identified that telomere lengths are highly associated to race, of course, age, and ethnicity and
Excellent.
sex.
And what population did you use for your study?
So in our study, we used the UK Biobank data set, which is a prospective large population-based cohort to be able to analyze both the leukocyte telomere length and the outcomes. So we followed the individuals for a median of 12 years, and we stratified individuals at their baseline leukocyte telomere length into tertiles. And we did the same with the Brain Care Score, which is a lifestyle modifiable risk factor tool that contains 12 modifiable risk factors. It includes social, lifestyle, and physical components. So we were able to calculate the Brain Care Score at baseline for all the UK Biobank participants. And that way we were able to stratify them into high Brain Care Score, which reflects healthier lifestyle choices, and a low Brain Care Score.
Can you give us a sense of the magnitude of impact that short telomere length had on your composite endpoint of stroke, depression, and dementia?
So in our study, after stratifying the individuals into leukocyte telomere length tertiles, we identified that individuals within the shortest leukocyte telomere length tertile had an 11% increased risk of developing the composite outcome of age-related brain diseases.
Is there anything an individual could do to mitigate that increased risk?
So our findings, when we put on together the leukocyte telomere length and the lifestyle choices, we do show that individuals that have healthier lifestyle choices compared to the ones that have a less favorable risk factor profile, individuals that have this healthier lifestyle choices are associated to a non-increased risk compared to individuals that have this unhealthier or less favorable risk factor profile, in whom we do find that they are associated to an increased risk of age-related brain diseases.
That's very encouraging.
Yeah, and additionally, we wanted to prove causality in our study to see if leukocyte telomere length was a causal or it was only a bystander associated to stroke, dementia, and late life depression. And in our study, we identified that leukocyte telomere length serves more as a biomarker reflecting other cellular processes that are happening within the body that might be the ones that are actually driving the association between stroke, dementia, and late life depression. So the telomeres are like an overall picture of how the body is doing. And in short, it's never too late to take care of your brain.
Do you plan to do any additional studies in this area?
Yes, of course. So our lab is very interested in other biological aging markers. So we do plan to try to do other studies that include other biological aging markers and try to develop a tool that can better predict individuals that are at highest risk of developing stroke, dementia, and late life depression to develop personalized therapeutics and interventions for these individuals.
Dr. Kimball, thank you very much for taking the time for this interview and for presenting your research at the International Stroke Conference in Los Angeles.
Thank you. Thank you very much for having me.